a great pleasure being here. I would like to thank the organizers for inviting me, especially Mehrdad, because I'm not working on barcoding. Mehrdad also told me I should go over time, that he has to talk less. I try not to do that, but if I do so, I blame him. So what I also don't work on, and I always say this in the beginning, is dinosaurs, because DNA doesn't survive in dinosaurs. Dinosaurs are way too old. Their remains, they went extinct a long time ago. We can't get DNA from. I found the talks I've heard here absolutely fantastic, although I'm not working on barcoding, and especially Paul's metaphor in his first talk about we are burning the books of life. I think it's a very powerful metaphor, and he's not right that people don't burn books, but what we know from history is if people do burn books, it's usually not good times. Well, I'm not working on burnt books of life. If they're really burnt, you can't do anything with them anymore. What I work on is kind of degraded books of life that have degraded over time. And for a long time, that was actually a very tedious way of working. We got very small data sets with a lot of work, and the interpretation of these data sets was very often rather questionable. This has changed completely during the last couple of years, and we're now able to look at complete genomes. And people do that. Svante Pääbo recently announced he's going to do 100 30,000-year-old human genomes to 30-fold coverage. That is something we could not have dreamed of a couple of years ago. And even a small lab like mine can do a few dozen low-coverage genomes from various species. Now, I should point out that in a way what we do are what Mark has called edge cases. So we're not processing hundreds of thousands or even millions of specimens. We're processing few specimens, and we are very much interested in how to get the maximum of DNA out of these and to look more into the evolutionary history. We do occasionally barcoding-like studies, but there are not that many species we can look at with ancient DNA for a start. So I'll mainly talk about technology and what has made it possible that we can look at complete genomes from degraded DNA. And there are a couple of things. Of course, there's the next generation or massively parallel sequencing. But for ancient DNA, there are other things like targeting the right part of the genome, improving extraction methods, which I think is also interesting for people who try, for example, wet collection samples or formalin preserved samples. Same is true for new library building methods and also for DNA hybridization capture. And I'll go basically through all of these, more or less in the order they're shown here. And I'll start with DNA sequencing revolution. I mean, nowadays I think most people are familiar with that. Mostly people use that machine, the Illumina in various versions. We have a NextSec, a very nice machine, also you can't get too high throughput with it. And what it has made possible is a massive increase in sequencing throughput, about 10 to the 7, 10 to the 8 orders of magnitude since we changed from Sanger sequencing to first 454 and then Illumina sequencing. And Mark has just shown a logarithmic scale as well, and I would think human brains can't work with logarithmic scales, so I'm showing this differently. This dot shows you how much data you could get from a single Sanger sequencing machine. This shows 454 sequencing, and if you take Illumina sequencing, it fills the entire wall. And I think this is fantastic. I mean, there are people who claim it's too much data, we can't handle it. I think that's nonsense. It's absolutely fantastic. The more data, as long as it's good data, we can get, the better. Unfortunately, in ancient DNA, this alone doesn't help us. It helps us a lot, but it's not enough. Because if we look at ancient DNA samples, this is from a study already 10 years ago and was actually done with cloning and sequencing clones. You can see that these are two different samples of cave bears that if we're lucky, about 5% of the sequences we get are cave bear. If we're unlucky, it's 1% or less. And the rest is bacteria, fungi, who knows what, maybe unicellular eukaryotes that have been growing on the bone. And so we can't really use these data. So that means if we take that sample, sequencing a cave bear genome costs 100 times of sequencing a modern brown bear, and because our sequencing reads are also shorter, the cost relationship is rather 600 times. That's actually very bad. So you can get 600 modern genomes for the price you can get one ancient genome. In a way, this has been known for a long time, because in 1996, somebody used hybridization, dot plots, to show that in that sample, a groundsel is actually only 0.1% of the DNA went back to that species. So we've known that for a long time, and embarrassingly, we basically didn't do anything about it. Also, we could have known there's something we can do about, because it really creates a problem of costs, unless you're really rich, which some labs are. And the point is, for a long time we said, it doesn't matter which bone you take, we just took what the curators gave us. But that was, of course, not the best approach, because there are differences in bones in the skeleton. And one difference between different parts of the skeleton is density. The densest bone in the mammalian skeleton is the petrous bone, and it's called that way because it's hard like a stone. And it yields absolutely fantastic amounts of DNA. Though this shows our current result on 23 petrous bones, and here you can see the endogenous DNA content. You can see it goes up to almost 80%, which is almost as good as modern DNA. And then it goes down. I mean, there are some samples that are really in the low end. These are very often very old, but basically everything is above 10%. So this is brilliant. And of course, you can't always have petrous bones, but what you can do is you can scan and get the hardest part of a skeleton. Or if you have other organisms, you can put some effort in figuring out where you get the best DNA out. In ancient DNA, we didn't do that for a very long time. And if you can't get any good samples from a skeleton, there's something else you can do. It was published in the last issue of Biotechniques by a former PhD student of mine, Matthias Meyer. You take your bone powder, you bleach it, and that removes most of the exogenous DNA. You get kind of variable results, but if you try it a couple of times, you can easily get up into the 50% range, which means apart from the problem of the sequencing length, it's only twice as expensive sequencing an ancient genome than sequencing a modern genome. There are other things you can improve, and again, this is kind of embarrassing for the ancient DNA community, because down here you can see a method we have used for a long time to extract DNA, and you can see how much DNA we got out here. Up here is a method that was published in 2013, and we get something like 40 times more out, so almost 2 orders of magnitude. And not only that, you can probably not even see it. We can see here that it's between 100 and 300 base pairs what we get here. And here you can see a lot of much shorter fragments with this method. And again, that is something we have known for ages. This is a paper from 1988, short but faithful pieces of ancient DNA. So already then, people knew that ancient DNA is short, it's fragmented. Not really how fragmented it is. That was done step by step. In 2004, we published a paper where we showed the shorter you go, so with 175 base pairs, you don't amplify anything. Then you can amplify 2 specimens. And the smaller you go, this is pretty short for PCR, the more you can get. Hendrik Poyner, in 2006, did a quantitative study, quantitative PCR, and actually showed that you get an exponential decay of copy numbers with increasing lengths. So you get 350,000 here, you get 1,000 here. And then finally, in 2010, we had the data from next generation sequencing, and it actually showed, if we go back here, the shortest fragment here is 84 base pairs. This is actually here. Here's 100 base pairs. That still means that you lose like 95% or more, 99% of your reads. Almost everything is in this very, very low range. Yet, the methods we used for extracting DNA were not fitted for very short reads, which is shown here. This is a comparison of the old method in light gray to the new extraction method that was published in 2013. And you can see here you only get 20% of your reads, of your DNA out of your samples. The rest is lost during purification. And this is actually also interesting for museum specimens, because we tried a combination of methods, including this purification method, and we got something like 160 nanograms per microliter of DNA from museum skins, so that's pretty good. So for us, museum skins are things we can do when we're fed up with our really ancient stuff. They are much easier. And we need these methods because if we go to very old ancient DNA, that means 150,000 years, what you can see here is that basically everything is below 60 base pairs. So the peak we get here is around 30 base pairs. So extremely fragmented DNA. Now that also means we have to think how we build our NGS libraries. This actually, I think, is a university library somewhere I grabbed from the net. And there are two ways. The way basically everybody uses is double-stranded adapters are added to your template. That's a classical Illumina sequencing library construction. Again, Matthias Meyer developed a different methodology based on single-stranded DNA. So what you do is you denature your DNA simply by heat. So you have single strands, and then you add a single-stranded adapter, bind it to a streptavidin bead, and then actually extend with a primer, add a double-stranded adapter on the other hand, and then you get your library. And some people have complained about this methodology and say it's not much better than double-stranded DNA. We haven't developed that, so that was long after I had left the lab. But I can tell you we implemented it and we get conversion ratios of 50 to 60% of the DNA you have in your sample. So you still lose half, but with the double-stranded you lose 90 to 95%. That's totally fine if you have enough DNA, but if you don't have enough DNA like we have, we prefer that one. And these methodologies together, they have made it possible to extend the time scale from what was believed to be the limit, about 100,000 years for ancient DNA, to 400,000 years outside the permafrost and 700,000 years inside the permafrost. Outside the permafrost, we might have shifted the limit to about 500,000 years, but we're not too sure yet. So that also tells you why dinosaurs don't work. They went extinct 65 million years ago. So 2 orders of magnitude deeper in time. It's not going to happen. These methodologies also have an effect on the DNA recovered. And what is shown here are 4 samples that were extracted with 3 different methods. And then the red ones are extracts that were converted with the double-stranded library construction, the blue ones with the single-stranded library construction. And what is shown on the x-axis is actually the average read length and the distribution. And you can see it's massively different. It actually differs more between methodologies within a sample than between samples across methodologies. And to show you that more graphically, this is the same extract turned into a library either with double-stranded or with single-stranded, and you can see that the read length distributions are massively different. If you then add a different extraction method, different purification method, you get pictures like this versus this. Absolutely massively different, and if you then want to reconstruct parameters like DNA fragmentation, you get completely different trajectories, and if you estimate DNA half-life from these trajectories, you get completely different half-life times and maximum survival times for ancient DNA. So the methodologies are really crucial in ancient DNA, and they are still changing. So we hope still to shift a little bit, getting more short fragments and more converted. Okay, so that's the methodology. Back to the endogenous content. Even with bleaching, with Petrus, sometimes you can't get over this problem. Sometimes you have samples that don't go over 1% endogenous DNA content. Sometimes you also don't want to do shotgun sequencing. And if you have a distribution like that, that creates a problem because if you don't want to shotgun sequence, you somehow have to target your DNA of interest. Now, if your longest read is about 70 base pairs, you're out of luck for PCR pretty much. You can do PCR that is that short, but given that you have 40 base pairs primers, you have 30 base pairs of informative sequencing lengths. Even the regular barcodes you use, you need something like 30 PCRs to get the lengths together. That's a bit unhandy, and if you want to do longer sequences, it's not what I would recommend. What we use is DNA hybridization capture instead. And it works in our hands absolutely beautiful. It's very simple. The idea, you have a known sequence. You have a synthetic oligonucleotide. You can actually even make your baits from modern DNA. And you have your library with your adapters. You wash it over, you hybridize it, wash everything away that doesn't fit, and then reload the stuff and sequence only what is complementary. Works absolutely brilliantly, and we are applying it in many projects. And I want to talk about one that is not really ancient DNA, but it's more related to barcoding, and that was on Riversharks. The Riversharks are quite interesting. There are very few specimens in the museum, and they're all of them very old, up to 173 years. And with PCR, we didn't get anything, so just no amplification. So we decided we try hybridization capture. So why Riversharks? They were actually thought to be extinct until 1997, when they were rediscovered in Borneo and Australia. And we wanted to compare these type specimens, so these were the type specimens for two species, to the newly discovered specimens. So we did mitochondrial DNA, and we did the complete mitochondrial genome, because it's not more work to do that with NGS and hybridization capture than doing a short barcode. And we got about 10 million reads for each of these specimens, and full coverage of the mitochondrial genome at a couple of, I don't know how many times, couple of thousand times. We then compared these sequences with modern samples that had been rediscovered from various locations, Australia, Southeast Asia, and here India and Pakistan. And you have to think about a river shark. That's an animal that's up to 3 meters long. So it's not what Mark's working on. It's real megafauna. So you think it should have some morphology attached to it, actually, that allows identifying it. Well, what we found is that it's obviously not that easy, because there are 5 described glyphis species, Siamensis, Gangeticus, Fowleri, Glyphis, and Garicki. And we actually found that 3 of these are synonymous. They are all here very closely related. They're probably one species. But we found another species that is not described at all from Borneo and Bangladesh that's out here. And this is for the complete mitochondrial genome, 16,000 base pairs. We found 800 differences, which is approximately 5% difference. So that's pretty sure another species. So even for an animal that grows to 3 meters length, you find cryptic species occasionally, which I find quite remarkable. We didn't find 28,000 MOTUs, but we found 1 cryptic species. You can also use hybridization capture across wider taxonomic ranges. So you don't need actually to know your species, your target species that well. And it works surprisingly well across wide phylogenetic differences if you do it the right way. We started this project on the Riversharks is part of a larger project on shark and ray phylogenetics. And the problem with sharks and rays is there's one properly done genome, and that is the elephant shark or chimera. And from that, we chose 1,400 target genes from the nuclear genome. And then we tried how well it works on itself, DNA from the same species, and then 5 skates and rays and 7 sharks. And the result was really encouraging, because the worst sample we had, we still got 1,000 of the 1,400 targets. And we actually found that it works up to 40% sequence difference. So even if you have only 60% identity across your sequences, if you tweak the conditions a bit, you can get out your sequences of interest. So in a way, it works absolutely brilliantly. We have now more than 700 species captured with this approach. So a large part of shark and ray diversity. And with that many species, it actually shows that there are quite a few more cryptic species even in these large guys. At the end, 2 cautionary notes. So far I've talked all about success stories, but there are limits to these technologies. So DNA hybridization capture works up to 40% sequence difference, but there is a limit. And we saw that when we looked at this guy, Homocerium, to be precise, Homocerium latidens. So Homocerium is related to the Smilodon. It's a saber-toothed cat. It was fairly large. This shows a modern line. This shows Homocerium. So it was probably not too pleasant to come across it. And it actually died out some 12,000 years ago in North America. And when it died out in Europe is a different question, because people believed it died out some 200,000 years ago. But there was a finding in the North Sea in 2000 that was dated with actually 6 different carbon dates to 28,000. So when modern humans entered Europe, these guys were still around, might have made interesting encounters. So what we were interested in actually is, where the heck do these guys come from? Because the last fossil is 200,000 years old, and then we only have them in North America. So we tried to get sequence data from these, and there are no close relatives of Homocerium. So we initially designed baits that were based on modern cats, but then we were very fortunate that another group accidentally shotgun sequenced a Homocerium. They thought it's an American lion, the bone, and found, hey, they suddenly had a partial Homocerium genome. They also had a partial Homocerium mitochondrial genome. So we designed capture baits based both on modern cats and the Homocerium partial sequence we had. And that shows you the distribution. Again, you can see very many short reads, a few a bit longer ones, but capture actually prefers somewhat longer reads. So you bias your distribution a bit to longer reads. And this is the tree. And here you can see we have 3 Homocerium, 2 North American one we captured, one that was shotgun sequenced, and our North Sea one. And they nicely fall where they should fall, basal to all other phyllets. However, this tree is based on only 11,000 base pairs, not the 16,000 base pairs of the mitochondrial genome, because what we found is we had parts of our capture baits that were based on the Homocerium shotgun sequence, and part of our capture baits were based on a reconstructed ancestral sequence of modern cats, because that's closer to Homocerium in the tree than any modern sequence. And these areas were very poorly covered, and sometimes we had gaps. And we have new data now, and my students didn't allow me to show them because the results are too preliminary, that actually show that if the sequence divergence becomes too large, the capture breaks down, and you get these kind of troughs in your sequence coverage. So you have nice coverage, then you have a trough, and then it goes on. So it's not smooth. It suddenly breaks down. What we saw on a different project is that actually the more sequence divergence you have between 0 and 25%, the less coverage you get, but it looks kind of smooth, but at some point out here, it suddenly breaks off. So if you work with species that are too divergent, hybridization capture doesn't work anymore. The other thing is identification of species, where I want to throw out a cautionary note. And this data come from this species, Elephas antiquus. It's an extinct elephant from Eurasia that went extinct, people are not too sure, either 120,000 years ago or 30,000 years ago, probably 120,000 years ago. And we have lots of samples from this species. So complete skeletons, beautifully preserved. So, and paleontologists are absolutely sure this is an elephant species, an Asian elephant, not an African elephant. We got from 2 different sites complete mitochondrial genomes, and we got a tree that looked like this. You have here the mammoths, you have the Asian elephant, you have the African forest elephant, the African savanna elephant, and you have Elephas antiquus in here as a sister group to the African forest elephant. Then we talked to paleontologists, and they outrageously rejected this possibility. Well, they actually said they must have hybridized, because elephants occasionally do that. As Mark mentioned, these are the edge cases. You have mitochondrial transfer. So we decided, okay, let's go for the complete genome. And so we got genomic data, not a complete genome yet, but when we use partial genomic data, the tree looks very much the same. So you cannot only find cryptic species with very large animals, you can also find completely incorrect genus assignments, and sometimes people won't believe you if you just present them with mitochondrial data. So I'm a very strong proponent of multi-locus data. I think it's very useful to have barcoding not only based on mitochondrial data, as great as it is, but sometimes I prefer multi-locus data. Okay, here I should thank the people who have done the real work, my research groups, and I was very pleased to see, I have this picture used for quite some time, that at the entrance of the university here, we have kind of a similar animal lying around. And sorry, Mur, that you have to give your talk. I'm at the end.